All right, welcome back to COG 266, Brains, Minds, and Consciousness. Today we're going to talk more about some of these laws of association, contiguity and contingency. So, last time, we went into some fundamentals of how we adapt our behavior to an understanding of the world around us. Habituation is how our responses to individual stimuli change over time, and classical conditioning is how we pair stimuli to produce new response patterns, right? So individual stimuli in the environment, and also a little bit about how they can be related from habituation, a similar response to a similar sound, versus classical conditioning is where we can pair concepts together, right? So pulling back to the laws of association, we have a little bit of resemblance going on there. We have contiguity, and then we also have this more interesting contingency and causation thing, which we'll get into shortly. Classical conditioning and contiguity, right? This was what we addressed last time to an extent. It needs contiguity to work, right? The conditioned stimulus has to come very close to and just before the unconditioned stimulus in time. If I ring a bell 5 minutes before a dog gets food, the stimulus pairing will never actually happen. I never say never, right? But it will be very difficult to form that connection, right? They have this temporal contiguity. They co-occur in time. Remembering this, remember in early trials, you present the conditioned stimulus a little bit before the unconditioned stimulus, and then the unconditioned response occurs, right? The responses are on the top, the stimuli are on the bottom. Early trials, you present early on, and all you get is the unconditioned stimulus. But later on, as you start to present that stimulus early, the conditioned response starts to occur before the unconditioned response, right? So present the bell, and then the food creates the salivation, right? But as you continue to do this, as they co-occur in time, the actual response patterns show that the response starts to creep into the period where there's just the bell has happened, right? And then we can remove the unconditioned stimulus and it'll occur, right? So this contiguity is very important to how classical conditioning works. Things gotta go together, right? If they don't really go together, what's it look like? Well, we can stretch it to figure out, what happens if we delay that unconditioned stimulus, right? How far can we push the limits of what is contiguous in time? At some point, it just stops being temporally contiguous, right? If I present a bell 6 days before I offer a dog food, there's no chance that they will pair those concepts together. So when does that stop happening, right? We know it works when it's like a couple seconds before. How far can we push it, right? On the other hand, it can't be literally simultaneous because your system has to have some time to start processing these stimuli. Otherwise, if I ring the bell literally at the same time as I offer the dog food, they're going to be way too excited about food to care about the bell. It has to be this window of these events occur together, right? So, long delay conditioning, if you extend that presentation delay enough, at first, that conditioned response occurs with the conditioned stimulus as normal. But in later training, let's look at this first. If you do this, if you have an extended delay, right? You ring the bell 2 minutes, 3 minutes before the unconditioned stimulus. Don't quote me on the minute amount. I'm just sort of illustrating the concept. The actual amount of time will probably vary with different stimuli. But the idea is that you have an increased delay, right? When you start this conditioning, it works, right? It will occur and it will sort of connect, right? So you train the dog up to salivate when I ring the bell early. But an interesting thing, you don't really need to salivate if it's a consistent delayed stimulus. You can actually start to see that later in conditioning, that conditioned response is also delayed to be closer to the unconditioned stimulus. So if the dog knows that there's going to be food in 5 minutes, they don't bother salivating early. The system wouldn't worry about that. It would say, food's coming, I'll get salivating later, right? So this is a phenomenon, our systems kind of trend towards co-occurrence, right? Now, on the other hand, simultaneous presentation, if you present them at the exact same time, that conditioned response rather, is going to be weak or non-existent. In fact, the system won't, your neurons, your doggy brain is not going to be able to form this connection, right? You are connecting really only to the actual stimulus to the response, right? You are just seeing food and there happens to be a bell. They are not triggering each other. So there is this ideal delay for conditioning. The exact delay time, it's going to depend on everything, right? Because some things are matters of milliseconds while other things are matters of minutes, right? But here's an example. When you're attempting to condition adult humans to produce an eye blink response, in older adults, a 1 second delay is ideal. In younger adults, a 0.4 second delay is ideal. So there's this delay period where if you wait too long, it's not ideal, and if you do too quickly, it's not useful. Now, here's another question. What happens if I try to have a conditioned stimulus after an unconditioned stimulus? Again, nothing. But why? What's the difference? Why is this co-occurrence in this particular triggering order matter? Why do we need to have a particular order to this? We're supposed to be pairing ideas in time because they are related to each other. Why can't the bell and the food be presented at the same time? So, it's because it's not just contiguity happening here. If contiguity were the full explanation of conditioning effects, we might expect simultaneous presentation to be the most important, right? But the experimental data are showing something else. Contiguity alone, again, so the model here, would predict that, hey, there should be no delay. All connections should be formed at the same time. As long as things are presented together, we know that they are connected, right? But the actual results are more like this. The triggering event has to occur a little before. The predictive event has to occur a little before for us to build something out of it. We can take an example of this, or take this question apart by looking at temporal conditioning. So a little bit of a different spin. No conditioned stimulus at all, actually. Just present unconditioned stimulus at regular intervals. I give food every 10 minutes. After some training, a conditioned response starts to occur based entirely on time, just before the stimulus is supposed to show up. So a stimulus appears, food is offered, and once they get used to the rhythm of this, they can start to learn to salivate early, right? They know food's coming, they're excited, their mouth is getting saliva-y, right? And okay, and remember, if it's been a minute since last time, it's not just saliva and food, right? All stimuli can be paired in some way. You hear thunder, or you see lightning, you know thunder's coming, right? Those are somewhat distinct events, but we know they are connected. So if I see lightning, I begin to expect thunder, right? That is a conditioned phenomenon. So if you have a pet, the difference between those two things is huge. They don't care about the lightning; they are terrified of the thunder. So this is the idea of contingency coming to play, right? Temporal conditioning effects show that conditioning can occur even without a paired stimulus. So what we're seeing is prediction is key. The law of contingency and causation is what we're entering into here. It's not just paired together, it's predictive power. And remember, that's very much like what we've talked about with hypotheses, right? Being able to test hypotheses in the world, right? So, continuing to interrogate these questions, is contiguity necessary for this to happen? So, if causation is part of the equation, how much contiguity do you actually need? Do we need contiguity, or is it enough that events just precede each other? Trace conditioning, another kind of conditioning that was developed to investigate these kinds of questions, can help us answer that. In trace conditioning, well, in normal conditioning, the stimuli are going to overlap, right? The conditioned stimulus, the bell, and the food are presented together to some extent. But trace conditioning is where you can use it as a signal instead. The bell can signal food, right? And people have to rely on a memory trace of this conditioned stimulus to make the pairing. It works similarly to normal conditioning, but it's a little bit weaker. So there's no, the stimuli are never presented together, but you can actually still form the association. So contiguity is not strictly necessary. The things don't have to overlap perfectly, though they do still have to in a sense, be nearby, right? Nearby in time. So, contiguity and contingency. The various forms of conditioning chart this middle ground between the conditioned stimulus and unconditioned stimulus overlapping, and the conditioned stimulus and unconditioned stimulus not being related at all, right? So this sort of space between where most of the actual associating occurs. So this implies that contiguity is necessary, but not sufficient for conditioning to occur. But predictiveness is more consistently important in all of these cases. So contiguity helps, but predictiveness is more important. So the role of contiguity is that it's kind of a prerequisite for even detecting contingency, right? If the stimuli are closer in time, there's a stronger conditioning effect, but it can't be backwards or simultaneous. You have to one stimulus has to signal the onset of another one. So helping to make these predictions, right? Remember that the brain or the mind is, in many senses, a prediction engine. So, prediction. We're seeing how conditioning seems to signal the importance of both of these laws of association, right? And we can see that contingency needs some contiguity to get the job done. But why is prediction such a strong focus? Why is it useful? What kinds of behaviors can even be predicted using this? So, we can talk about the prefiguring hypothesis. This is an attempt to try to explain conditioning's connection with prediction, right? So what it states is, classical conditioning optimizes interaction with an upcoming biological event. So unconditioned stimuli are typically innate reflexes related to biology, right? The bang means danger, good smells mean food, knock on my knee means, react, right? Predicting biological events is the idea that, so given any stimulus that predicts food, for example, an organism that is able to adapt itself to seek out that stimulus, as well as food, is going to have a much easier time navigating and surviving its world. What this is getting at is that there are regularities in the world, right? I see a certain kind of tree, I know it has good berries on it, even if I can't see any berries right now. Detecting the berries is one thing. Detecting the possible presence of berries, predicting the possible presence of berries is another. And the idea being that being able to predict these things also helps you prepare yourself for these things. If you are able to use additional information from the environment, then you're able to be ready in a sense. Here's an example, a mouse mating study. So if you pair the presence of a mate with a conditioned stimulus, so the blue mouse is excited by the presence of a pink mouse, right? Pair that association of the pink mouse's appearance to a sound, and the blue mouse is actually, it produces an increased arousal, right? So the mouse is even more ready to go, right? So again, being able to respond to these mating signals is useful, and it helps the mouse get prepared to be, I'm going to court. I'm going to do my mouse dancey things. I actually don't know what mouse courting is like, but please don't send me any videos. It's fine. But the concept is that this is a preparatory ability. Without the stimulus being able to predict the presence of the female mouse, then they just a normal amount of arousal. And that's fine. You don't necessarily have to have mice that are hyper aroused, but you get the idea that this is perhaps useful. If you know that, say, that pretty bird over there, its nest signals that it's here and ready, you can yourself get ready. Right? Similar, so we have mating in nature. We also have lots of fighting, territorial things. If you pair a rival fish's presence with an unrelated stimulus, the conditioned stimulus actually increases the number of bites and tail beatings in the signaled fish. So the fish who knows that an opponent is nearby is able to predict an upcoming fight and prepare itself a bit better. Not great, we're not trying to encourage fish violence, but the fact is that fighting is something that happens often in nature, right? So we're talking about food, fighting, and mating. Also, as a note, John Cena's music does not really help him win, since he's always signaling way too hard. Maybe he's just flexing. Who knows? Another one, fish foraging. So this is different, right? This is not about the fish fighting each other, but if you pair the stimulus with the absence of a rival, so there's a light on when there is nobody looking, the fish will be more willing to engage in foraging and nesting behaviors, things where it knows it doesn't have to defend itself. So if you're able to use the stimulus in the environment to make sense of what's happening in the environment in terms of very important things like, am I going to get my tail beat if I let my guard down, then that's useful. That is biologically useful. It's great. So, conditioning and biology. Classical conditioning seems to be useful in building up our predictions about things that affect us biologically. But what's the goal here? So, like, why would a system arrange itself around these things? There's sort of a basic pattern that we're seeing, like, oh, it's nice to be able to prepare, sure. But what is driving this evolutionarily? And that is the optimal time allocation hypothesis. So, alongside the prefiguring hypothesis, conditioning can also help us to optimize our time resources. Something that you should probably internalize as a person, as an entity, as a biological agent, is that time is the only resource you actually own. You might think that you have all these access to money or whatever, like as you progress through life, but all that is just time, baby. Money is a representation of work spent, right? So time is key to survival, to our existence, to life, to everything that we do. So time is a very critical resource. And every useful activity takes time and effort for an organism to commit to. So being able to understand signals in your environment, i.e., maybe I should develop a neural system capable of doing prediction, then, hey, help me stay situationally aware and develop habits. So now even larger patterns can emerge, all in service of using our time wisely. So habits like, if I can predict when and how other predators, or rather potentially just rivals, move in my environment as a fish, then I can kind of start planning a route for my day, you know? So How do we identify what's important, though? We have this phenomenon, we've discussed this conditioning phenomenon with the idea that, oh yeah, if I can predict things in my environment, that's great, that's beautiful, that's important for me to be able to do. So if I see a light bulb light up, I'll know that Jerry's not coming to beat me up. Cool. But there's a lot of information in any given environment, right? That's not just a light bulb and a mean fish. It's sounds, it's everything. Initially, you can imagine a world where we have no idea what signals anything at all, right? All events are equally likely in our minds because we can't predict anything. We don't know anything about the world. But we gradually piece together which stimuli in the environment are actually signaling important events, right? For example, here's the world full of stuff. There's a mouse in a cage, it's got a lever, it's got sound and light coming in from outside, there's a clock on the wall that makes a sound sometimes and it has a visual stimulus, there's a person who comes in sometimes with food, there's a door that can move and make sounds, blah, blah, blah. There's so much stuff in the environment. How do we actually identify the causation, right? There is always a little bit of sound coming in from outside. Shouldn't that just make me think that all outside sounds mean food? This highlights the problem of projectable predicates. I love our alliteration in these sciences. So, predicate is a necessary condition for something to occur, right? A cloud is a predicate of rain, right? Projectable means that can be chosen. So this is the problem of induction, problem of credit blame assignment, all these things together, kind of the general idea. How do we figure out which things are responsible for which other things? So here's kind of the problem. Given two equally likely things, unconditioned stimuli, for example, a light and a sound that could signal food, which one is the one that receives the new association? Initially, it may seem like we have nothing to go on. If I presented all of what we talked about in terms of classical conditioning to you, you might just say, well, I don't know, 50-50, right? Maybe both. It has to be both, right? But again, it's about probability structure. So they have the same chance of occurring. They're both 50-50 shot. Which one receives the association? Well, for one thing, all things being equal, and the same presentation time and all that, the more salient a thing is, so the stronger and more noticeable it is, that will overshadow the weaker stimulus. So overshadowing is one method. The things that are most noticeable in our environment, i.e. things that maybe just recently changed, like the door is suddenly open, that's important. A change is detected, and we know from neurons that changes can be detected pretty easily. Stronger stimuli, louder sounds, or a faint light versus a extremely loud sound, or a very bright light versus a very minimal sound. Again, there's the concept of response response strength, right? These are ways that we can actually help select. Here's another one, blocking. This one's interesting. If one of the stimuli was previously associated, it wins. So there is food and there is sound with the food. Do the sound, get salivation. Next time around, there's food and there is sound with a light, potentially. The light actually gets omitted because we already know this association. Why would we care? Well, the theory is that there's no new predictive information. We are still able to use the old association. I am still able to know that, hey, sound is the thing that's been working so far. Why would I jump off of that right away? Then there's equipotentiality. Ooh. So two stimuli that have the same probability, same salience, same informativeness, okay? what if they really are identically predictive? Well, an interesting hypothesis here is, who cares? Both stimuli have the same utility. Pick one at random and get on with your day. That is not a very satisfying scientific hypothesis, but it's one that your neurons would probably go for, right? Why care? You know, if we've constructed this very structured environment where there's things that are perfectly equally distributed and they are both perfectly equally signify the presence of food for the dog doesn't care. It'll pick one, right? But that's given that all things being equal, you know, that's the predicate here, right? Things must all be equal for that randomness to happen. In fact, there are certain things that our systems are primed for. Some things are just primed more likely to be associated with other things. All things being equal, some stimuli are just more pairable. So, for example, taste aversion learning. This one's big. This is one of the only things that you get one-shot learning in. So, pair both a light and a flavor stimulus with either a shock at the end of a short delay or radiation that makes them sick at the end of a short delay. Yes, it is sad to hurt the mouse like this. So, again, pair the mouse. It will experience the light or the flavor. There's radiation at the end of it. That's case one. Case two is same thing. There's either a light or a flavor, and then they get a little shock. Here's the deal. If you present just light, those rats who were shocked after the flavor and the light together, they show avoidance. They're not going to drink the water. The water shocked them, but they're not picking up on the flavor. In fact, if you just show the flavor, only the irradiated rats show more avoidance. They have paired something that is more likely. So, let's get why. Why should this be? Why are visual stimuli, like the light, more associated with shocks than flavor being more associated with illness, right? You're not going to pair things like light with illness. Light doesn't typically make you feel ill, whereas flavor doesn't typically make you feel external pain. This is called preparedness. So organisms have this predisposition towards certain associations. Why might a rat be more ready to associate illness with flavor? Let me think about that for even half a second. Yeah, it makes sense, because they are primed to be able to avoid noxious foods in the world. Eat too much of that, you die, right? And then shock with visual stimuli, same kind of situation, right? You're not a animal, a rat that sees something startling is going to be potentially expect pain in that scenario. Now, here's an interesting wrinkle to this. Study by Wilcoxon, Dragoin, and Crowell, 1971. Why do rats and quails have opposite patterns in this preparedness? So, rats have what we just discussed. They are more ready to pair pain with visual stimuli and more ready to pair food or flavors with illness. Quails are the opposite. Quails are more likely to pair flavor with pain and visual stimuli with illness. The difference is rats are nocturnal and they use scent to forage things. They use sensory flavor chemical-based senses to detect food, right? Whereas a quail is diurnal. It's going to use vision because it's a forager. It cares more about what things look like to determine if they're good food. So if say it eats a certain kind of, insect that is a bright color, and oopsie doopsie, I got real sick from that. It's not about the flavor for the bird, but it's absolutely about flavor for rats. So this is an interesting look at the fact that different species can be differentially prepared for these kinds of associations. These relationships are specific to species, environments, other factors, right? But what determines preparedness? What kinds of associations can an organism pick up? Is it just genetics? Is it just the environment? Is it other more complicated factors? Maybe. Maybe. The fact is, it's not always clear, and I can illustrate how unclear it can be with some interesting mysteries. So, preparedness. Here's an interesting one. Chick pecking. Within 2 days of hatching, chicks will peck at mealworms. Yay, eat these worms. Good. Good for chicks. But if you cover their feet, they stop. Why? I don't know. It's not completely clear. There is some kind of neural triggering happening where their feet being covered is an important signal to their systems that they should not be pecking. But we're not sure why. It just happens to be. It's likely a combination of the very complicated things going on in a neural system. Here's another one. Snake aversion. Monkeys fed only fruit have no fear response to snakes. Monkeys fed fruit and insects have a fear response to snakes. Fear of snakes is pretty important, right? It's a useful thing to have, but for some reason, if you feed monkeys no insects, they're not afraid of snakes. Again, That's okay. We don't always know the answers to these things. But this leads to that question of, what the heck does a chick's feet covering have to do with pecking worms, and why do insects seem to influence unrelated fear responses? The answer is, and it's not a satisfying one, sorry, welcome to cognitive science, we don't quite know yet. It's complexity, right? Oftentimes the connections between innate predispositions, right? Developmental events like what you've been fed as you grow, and perhaps what you've experienced while feeding, from insects, for example, and the associations between stimuli, they all combine in these complex ways that they aren't clear. It's not clear how all those things interact to us, because dang, there's a lot of moving parts. In fact, when you think about the model of how neurons work, every neuron is a moving part, you know? It's huge. It's more complex than political science. But the point is that this complexity makes a lot of these kinds of interactions not very obvious. We're once again in a space where it becomes difficult to explain the machinery of the mind, of the neural interactions here, because it's not obvious. It's hard to trace these things. So, to recap, we're seeing that conditioning is this way to create new associations, right? It seems primarily aimed at detecting at least contingencies in the world, cause and effect, this sort of hypothesis testing, this prediction testing. But it does require a bit of contiguity. Things have to occur together. Some stimuli are more or less likely to be associated as signaling an event. So preparedness, salience, previous learning, when certain things are preferred, right? When I taste something gross and I feel very sick later, yeah, that's an easier connection to make. So, classical conditioning, we see that there's some variation within classical conditioning, but generally it's a useful way of understanding how we can associate ideas. Importantly, no single theoretical construct is capable of accounting for all aspects of classical conditioning. Contiguity, contingency, informativeness, evolutionary purpose, etc. These are all needed to make sense of classical conditioning. It's not just a straight up simple law that you apply everywhere. You need lots of angles to make sense of how these phenomena happen. But ultimately, it's a fairly neurological process that can really help to explain a lot of complex phenomena in our minds. Very cool. So that's it. As always, a final thought. What's a stimulus from your daily life that you use to signal the presence or absence of something useful to you in a biological sense? What about in a less biological and more psychological sense? What are the commonalities of these associations? What are the differences? All right, that's it. Thanks. Take care.